Mechanism of enzyme action. Hydrolysis of sucrose to glucose and fructose when done without enzyme requires 26,000 kilocalories of energy, and with enzyme sucrase it is only 9,000 kilocalories. This is because enzyme facilitates sucrose to be placed in a high energy transition state followed by its conversion to glucose and fructose. A variety of models have been proposed to explain mechanism of enzyme action, and these include Michaelis-Menten theory, which states that enzyme combines with substrate to form enzyme substrate complex, which is a reversible reaction. This is followed by dissociation of this complex to form enzyme and product. This is followed by Fischer's template theory, which states that substrate fits into the active site of the enzyme just like a rigid lock and key. Now, this theory failed to explain the attachment or association of the structure of the substrate with the active site of the enzyme. Now, this drawback was overcome by Koshland-induced fit theory, which states that the binding of the substrate to the active center of the enzyme facilitates conformational change in the enzyme so that appropriate binding takes place. Now, with all these models, it can be concluded that substrate binds to the enzyme, and this induces conformational change in the enzyme, which brings about appropriate orientation of the catalytic groups at the active site of the enzyme. Now, this facilitates appropriate and optimal binding of the substrate to the enzyme. Now, this will be followed by variety of interactions occurring between the substrate and the enzyme at its active site. Now, these interactions include the acid-base catalysis, covalent catalysis, substrate strain, product substrate orientation, and catalysis by metals. Now, coming to acid-base catalysis, now the good example for this is the breakdown of ribonucleotide, which is made up of base, sugar, and phosphate by ribonuclease enzyme to form ribonucleoside, that is base sugar with release of inorganic phosphorus. Now, this ribonuclease enzyme has got histidine at its active site, and this is present at two places, at position 12 and position 119. At 12 it acts as proton acceptors and it exhibits attack at the 2' position of the ribose sugar, whereas at position 119 the imidazole group of histidine acts as acceptor of the proton and cleaves the inorganic phosphorus from the ribonucleotide. That is how the acid-base catalysis is brought about by the imidazole group of histidine in the ribonucleose enzyme. The next one is covalent catalysis. This mainly involves a transamination reaction where the non-essential amino acids are synthesized in the body. Now, this transaminase enzyme requires PLP, pyridoxal phosphate, as the coenzyme. Here, the aspartate combines with alpha-ketoglutarate, the catalyzed by transaminase to form oxaloacetate and glutamate. Here, glutamate is a non-essential amino acid which is synthesized from aspartate. Now, the covalent catalysis is as follows. Now, the main group transfer is mainly done on this pyridoxal phosphate. Now, the aspartate transfers the amino group to pyridoxal phosphate, that is aldehyde form. After accepting the amino group, the pyridoxal phosphate gets converted to pyridoxamine. Now, aspartate, after donating the amino group, gets converted to oxaloacetate. In the next step, this pyridoxamine transfers the amino group to alpha-ketoglutarate, so that alpha-ketoglutarate gets converted to glutamate and the enzyme with pyridoxal phosphate is released back into the cytosol for the next reaction. So the next theory is the substrate strain theory. Now, this is the example of digestion of glycosaminoglycans or mucopolysaccharides by lysosomal enzymes. Now, these mucopolysaccharides are made up of repeating disaccharide units, and this lysosomal enzyme converts the stable chair form of this disaccharide into unstable chair form, bringing about the hydrolysis of the disaccharides of glycosaminoglycans. The last theory is the product substrate orientation theory, which states that the binding of the substrate to the active site of the enzyme facilitates appropriate 3D alignment of the substrate to the active site of the enzyme, facilitating a good chemical environment for the conversion of substrate to the product. Now, so depending upon this specificity of the enzyme, the substrate is converted to product either by one of these mechanism, as either by acid-base catalysis or covalent catalysis or substrate strain theory or product substrate orientation or catalysis by Medtouch. So either of the one or combination of these interaction will facilitate conversion of the substrate to the product, and this is followed by release of the product, and the free enzyme is released back into the cytoplasm or the mitochondria.